And the first speaker will be Malcolm Burns, and he is head of the GMO Analytical Unit and principal scientist based at LGC. He is an expert in the identification, quantification of genetically modified organisms in food products. He is manager of the UK National Reference Laboratory for GMOs and also dealing with method validation. So one of the key topics, I think, also here for this conference. 60 peer-reviewed papers, consultant advisor to different branches of the UK government, and participating in international working groups and expert advisory committees. And his talk will be about analytical strategies for detection of GMOs and NGTs. Malcolm, the floor is yours.
Great, thank you, Ralph. As Ralph said, my name is Malcolm Burns. I'm the head of the GMO Analytical Unit and principal scientist at LGC in the UK. And I'm here this afternoon to give you a brief overview entitled Analytical Strategies for the Detection of GMOs and NGT Products, Status and Challenges. So a big warm welcome to everybody in the audience today and all of our online participants. In terms of the context of my presentation, I wanted to give you a little bit of background and context of LGC, its official roles and how it's related to GMO analysis before moving swiftly on to the main part of the talk, which is both the status and challenges associated both with detection of conventional GMOs as well as detection of NGT products and signposting to relevant conference sections and presentations as well, before finalizing with some conclusions on the road ahead. Okay, so before I start then, in terms of acknowledgements, as my predecessors said as well, welcome everybody to the conference. I think this conference does provide a unique forum for the technical and scientific exchange within the international community on opportunities and challenges presented by GMOs and NGT products. Many thanks to the organizers of today, including BFR, BVL, BMEL, JKI, the European Commission, JRC, SCBD, but also obviously personal thanks to Herman Broll and Ursula Vincent for extending the invitation. And an official disclaimer, a caveat as well. The views and thoughts and opinions expressed during my presentation today are my own personal ones and do not necessarily represent the views of the UK government or its associated departments. So moving swiftly on to the background and context of the presentation. LGC, it's a life sciences measurement testing and research institute its headquarters based in Teddington in the UK. We celebrated our 180th anniversary last year, and we currently operate out of 19 different countries, consisting of around 4,300 employees. We have a number of official roles related to LGC as well. One of these is a national measurement laboratory. So we're the UK's designated institute for chemical and biomeasurement, and we work globally to help harmonise measurement science. We also have the government chemist function at LGC, which is underpinned by the relevant UK legislation, and that is to provide impartial and independent referee analysis on a food sample as part of official controls in cases of dispute, for example, between a food business operator and a local authority. And as a result of the government chemist function for the last 3 years, we've had 12 GMO cases referred to us for analysis as part of this function. And finally, we also have the two functions of the UK National Reference Laboratory, the NRL for GMOs in food and feed, pursuant to the retained EU Regulation 2017-625. And we're also the GMO authorisations position in Great Britain, so provision of scientific method validation services as part of the official authorisation process. And these two final positions are awarded and funded by the Food Standards Agency. So we've been involved in a number of example guidance documents and publications and authorships on these, both in the past and present, actively supporting both the European Commission and the European Network of GMO Laboratories. We've got some examples there, the one on the left with JRC technical report on digital PCR by Sven Pecoraro. The middle one, an uncertainty measurement guide in its 3rd edition for GMO testing laboratories. I think if I remember correctly, Herman, the first one was done in about 2008, 2009 now by ourselves, others, and Stephanie as well. And an example document on the right there, which we've already seen in previous presentations on the detection of food and feed plant products obtained by new mutagenesis techniques as part of the ENGEL. Close to home in the UK, we also actively contribute to other publications, a genome-edited edition on food crops as part of UK Parliament Post, and also editing and authorship on a Royal Society of Chemistry book entitled DNA Techniques to Verify Food Authenticity. A number of authors already present in the audience today as well. Okay, so moving swiftly on to the main part of the talk, detection of conventional GMOs, status and challenges. Before we go launching straight into this, I want to try to put a little bit of a definition on there as well. At the minute, what I'm doing is talking about conventional GMOs. Now again, we refer to the EC JRC technical report by Wim Broodthaerts there. In terms of conventional GMOs, are organisms produced using recombinant DNA technology, typically containing DNA sequences randomly introduced from the same or other species prior to the adoption of Directive 2001/18 EC. And indeed, if we're talking about status and challenges, this is already one of the challenges. In order to harmonize the terminology, we need to think about this sort of going forward to create a level playing field and understand what all the terms refer to as well. So sometimes it's referred to as classical GMOs, but here I'm referring to it as conventional GMOs. Now, the whole aspect of control in GMOs, one of the underpinning parameters is supporting consumer choice and recognizing that consumers may not want to purchase food which contain GMOs. And within Europe, the traceability and labeling of GMOs is governed by the relevant legislation, which dictates that products containing GM material must be clearly labeled. So the successful labeling of a food produce is dependent upon the reliable, stringent, and efficient way of quantifying GM. So the largest common denominator in the global framework of GMO analysis is that of DNA-based methods. And the majority of accepted methods of GMO detection and quantification are DNA-based. There's a number of reasons behind that. For example, DNA being ubiquitous, whereas proteins as a target analyte can be expressed at a different developmental stage of an organism's life or be tissue-specific, etc. And indeed, quantitative PCR-based analyses is the current preferred DNA-based technique for routine GMO analysis, being very practicable. And indeed, the EURL validated methods for event-specific detection of GM varieties underpin this and provide unequivocal target identification. It's based on the unique integration region between the transgene insert and the host genome, creating these events as such. And again, if I can point everybody to the EURL there and the validated methods that they produce, there's a link at the top of the screen there. These are all underpinned by solid method validation, both intra-lab validation and also through organisation of collaborative international inter-lab trials. And talking about the rest of the conference, in session 4 we'll be learning a bit more on detection of classical GMOs from colleagues in the and participants at the conference today as well, including Frederic and Frank. So one of the other challenges of GMOs is the number of GM lines that we are being asked to detect. Now, as we learned earlier from Guy in the late 1990s, early 2000s, there was only a set number, a certain limited number of GM lines which were approved for use and placing on the market in the EU. And following the moratorium on GMOs, we were only talking about between sort of 10 and 12 GM events that had to be detected in the early 2000s. If we look at the 2021 there, that had risen to about 90 different GM lines, which in itself provides an operational challenge for analytical laboratories in order to detect all of these GM lines and events. So one of the ways to approach this is to do a GMO screening approach and to capitalize upon some of the common GM control elements or markers which are present during the normal transfection or transformation phases, such as some of the promoters or terminator regions there. And we've got two example mainstream approaches, which I've quoted here. One is based on the EURL JRC pre-spotted plates by Madalena Quercy, first published in 2009, which are lyophilized primers and probes, providing a very useful sort of hands-on approach. And if anybody would like to learn more about that, I know Madalena is present at the conference today. And also other matrix-based approaches as well, for example, by Weiblinger et al., whereby we're cross-referencing results from an informative panel of screening markers to known occurrences in GM lines. So therefore, knowing a priori what markers are present, you're able to capitalize upon that and therefore have a look at potential sets or subsets of GM lines that may be present in a sample. Now again, all of these approaches are very much dependent upon and reliant upon databases. And in session 6 of the conference, we'll be hearing a bit more on global information sharing. So we'll be hearing from Laura Bonfini, based on the EC, JRC, and the EURL GMF database, but also from Theo Prins as well on the Eugenius database. So techniques for GMO analysis, there's a number available, broadly categorized into three general sort of areas there. The first we've already heard about, real-time PCR. In terms of practicability, this is already embedded into the infrastructure of a number of analytical laboratories there, offering both excellent specificity and quantitative capabilities. Digital PCR is one of the new kids on the block, as it were. This is gaining increasing traction, but still slightly less common than real-time PCR. This can afford absolute single molecule quantification without reference to a calibration curve there. And again, if I can point everybody to the JRC technical report, authored by Sven Pecoraro et al. In 2019, that makes an excellent reference for that. And also apologies for the typo, next generation sequencing, not next generation sequencing, which can manifest itself in a number of applications in terms of massive parallel sequencing, whole genome sequencing, metagenome, metabarcoding, etc. The cost of supporting the infrastructure, the bioinformatics and the skill set may be quite prohibitive at the minute, but it's generally agreed that such things as next generation sequencing do provide good potential to identify unauthorized and unknown GMOs. And again, that's potentially the second main challenge that we are still facing today for conventional GMO analysis. And again, if I can point people to the relevant sections today, I think after myself, Alexandra will be talking a little bit about identification of known and unknown classical GMOs and NGT products. And in session 2 tomorrow and session 4, we'll be talking about looking at detection of NGT products and detection of classical GMOs as well. So to finish off then, GMO controls, the analytical framework to detect and quantify GMOs are based on three things, as explained by Hendrik Emmons et al. In his 2018 paper: knowledge of the altered genome sequence, availability of reference materials, or more importantly, certified reference materials, and also the availability of a validated detection method. And this is certainly underpinned by all of the work that the EURL has been doing in this area. So moving on to the second part of the talk then, detection of NGT products, status and challenges. Before launching straight into this, again, we've heard from previous speakers today about one of the pivotal publications in the field, the ENGL 2019 guidance note on detection of food and feed plant products obtained by new mutagenesis techniques. I understand this is currently being looked at and updated as well, but the actual conclusions still hold true for today. What do we mean by gene editing? Well, there's an ISO definition available, which refers to this as being a group of new targeted mutagenesis techniques that facilitate addition, removal, or alteration of DNA sequences at specific locations in the genome. Now, this is distinct from the original conventional GMOs, which can be defined as, which are underlined by creating being created by transfectional transformation, incorporating larger pieces of foreign DNA into the host genome, usually from outside the breeder's gene pool there, often at random sites, frequently alongside easily identifiable markers, so promoters or terminator cassettes. And indeed, gene editing can generate different kinds of alterations in the genome, ranging from single nucleotide variations to deletions and insertions of many base pairs. Now, today's conference really is focusing on some of the technical and scientific aspects, and we've had some general overviews of some of the key development dates, but of course, in July 2018, the European Court of Justice ruled that products of gene editing, synthetic biology regarded as GMOs, and fall under the pre-existing legislation for GMOs. The European Commission has since then done a range of studies and consultations, including one based on an impact assessment on NGTs, of which the public consultation factual summary report has already been published. Closer to home for myself as well, in the UK, in May last year, there was the Genetic Technology or Precision Breeding Bill. Now, this is a proposed legislative change in the UK, which is to bring forward primary legislation to amend the regulatory definitions of a GMO to exclude those organisms that have genetic changes that could have been achieved through traditional breeding or which could have occurred naturally. Now, at the current stage, I looked online this morning, it's in its final stages of amendments prior to royal assent by the King and approval. The regulatory and practical implementation of this bill is still being considered by UK governmental departments and related expert advisory groups. But of course, a future analytical challenge, similar to NGTs, will be to determine if any DNA sequence variation we see in a food or feed product could have arisen through traditional processes, natural transformation, or as a result of gene editing. So, one of the key challenges for detecting NGT products. First of these is, we've already heard about, technical detection of small sequence alterations. It's technically challenging, but entirely feasible to detect small genomic alterations, for example, single nucleoside variations, single nucleoside polymorphisms. Indeed, modern molecular biology techniques, inclusive of qPCR, digital PCR, and NGS, offer the best potential for detecting genetic changes in an organism's genome, given a priori information is known on that sequence and potentially the flanking regions to that sequence as well. And tomorrow we'll hear about some of the work being pioneered by Science Sano, Marie Alice, and Nancy Roosens there on the detection of NGT products. And should sufficient information be known regarding a sequence alteration, and confidence be attributed to that sequence alteration being specific to a GMO line, be that conventional GMOs or potentially gene-edited lines, then detection, identification, and potentially quantitation can be achieved. And that was in one of Lutz Groman's papers in 2019. So, whilst there is general agreement that modern molecular biology techniques, qPCR, digital PCR, and NGS offer the best potential for detecting genetic changes in an organism's genome, what they cannot necessarily do, when used in isolation and when targeting single mutations, they cannot necessarily provide you with unequivocal evidence as to the source of that mutation, be that either as a result of gene editing, natural mutagenesis, or traditional breeding approaches. Now again, we've got to look at this in context, and the previous presentations have also highlighted this. New genomic techniques is an umbrella term incorporating a diverse range of techniques which can differ both in the mode of action and the resultant mutation as well. So for example, if we're just looking at a single nucleotide variation, being able to detect that that has been as a result of gene editing may be very, very challenging. But in specific instances, detection may be possible if enough additional information is available to prove that a DNA sequence or sequences are unique to a specific gene-edited line. And again, an example could be that following some types of site-directed nuclease 3 activity, which can result in larger stretches of DNA being present in predefined insertion regions in a genetic locus. But as Emilio was saying, we need to look at this in terms of sensibility as well, practicability. What are the likelihoods of SDN3 related gene edits being present in the pipeline for those applications as well? As we saw earlier, the majority of these are actually based on SDN1 activities as well. So just rounding off then, other challenges for detecting NGT products. The terminology we talked about earlier as well, I briefed upon. It's an umbrella term encompassing a range of diverse techniques. But again, there's a range of diverse terminology to describe the processes and products, including established genomic techniques, new mutagenesis techniques, plant breeding techniques, new plant breeding techniques, etc. So it's useful to define and agree the terminology to promote a harmonized approach and greater understanding in this sector. And again, key texts are being produced by EFSA and also in the UK, DEFRA's ACRE committee, which is the Advisory Committee on Releases to the Environment. Also, quantitative estimation may be very important with respect to compliance with a legislative threshold. But the lack of agreement currently on detection and identification methods precludes effective approaches for quantitative estimation. Also, screening for NGT products. This may be very challenging in the classical sense due to the lack of inserted elements, which would be common as a result of gene editing activity. Some groups have postulated that it may be possible that methods detecting short DNA alterations without necessarily identifying the NGT product could be used in a very limited sense for screening. But again, a prerequisite is that the sequence of interest must be known beforehand, and also reference materials and comparators. Any analytical approach, the efficacy of any analytical approach, be that genomics, proteomics, etc., is completely dependent upon the availability of appropriate reference materials and/or curated databases. And again, because of the potential transient nature and segregation of multiple on and off target mutations, some agreements need to be reached in relation to what can be considered as an appropriate reference material. Databases as well, we'll hear about later in the conference in session 6 as well. Could be used as a very good comparator, ideally as a pangenomic database per taxon, but the practical challenges associated with establishing that are very real and very challenging. Again, I think in the States there's something called the Maze GDP, which is the Maze Genetics and Genomics Database, which is a federally funded initiative, and it's a informatics service looking at the genetics associated with the Maze genome. And finally, research into detecting products as a result of NGTs. This is the concept of a weight of evidence approach and minimum qualifying information, based on the concept of collective information gathering to build up almost a unique signature or footprint associated with an NGT product. So not just looking at the site of interest, but taking into account other informative aspects as well, including flanking regions, the genetic background, any linked off-target mutations, which show complete linkage and will co-segregate with the mutation of interest. Some groups have also postulated the idea of heritable epigenetic and epitranscriptomic changes, and also such things as blockchain and other documentary evidence, the supplier origin pedigree, etc. All of these, I think, offer potential viable routes, but there is a requirement to provide additional evidence-based experimental work to further support or refute some of these hypotheses. And again, we'll hear in session 2 and session 7 on the detection of NGT products and alternative approaches for traceability. So to finish off then, again, even for NGT products controls, it requires an analytical framework to detect and quantify GMOs, knowledge of the altered genome sequence is important, certified reference materials, and a validated detection method. And in session 3, we'll be hearing from Slavomir and Patrick a bit more on some of the requirements for the identification of those NGT products. So to conclude, on the road ahead, in terms of conventional GMO controls, and then in terms of conventional GMO controls, the analysis is grounded in evidence-based research and innovation, as pioneered by the European Commission, JRC, EURL, GMFF, and also as supported by the ENGL. Core molecular biology aspect is that should sufficient information be known regarding a sequence alteration and confidence can be attributed to that sequence alteration being specific to a GMO line, then detection, identification, and potentially quantitation can be achieved, but further challenges still do await with conventional GMOs in terms of the number of GM lines and the presence of unknown and unauthorized GMOs. And in terms of detecting products arising as a result of NGTs, there are significant challenges still in the road ahead, including technical detection of small alterations, qualifying the source of the mutations, screening, quantitation, etc. But all of these significant challenges are not necessarily insurmountable. And again, apologies to Guy, because this was the only photo I could find on the internet, and Guy has already used this beforehand. But at the ENGL inauguration ceremony in 2002, as Guy has already explained, there were still a number of issues associated with conventional GMO analysis, including such things as how do we validate a method? How do we verify that method is applicable in a laboratory? What about measurement uncertainty? What about conversion and expression units as well? But the ENGL, under Guy's guidance and vision, provided a unique forum to discuss and elaborate on those issues. And again, I do believe today that this conference also provides such a forum for the exchange of information and working together. And again, we'll hear from other experts in session 5 and session 7 on the regional networks and alternative approaches for traceability. I very much look forward to the rest of the conference, where we'll be hearing from international experts regarding their thoughts, insights, and sharing of experiences for detecting NGT products. Thank you very much for your time. Again, I'd like to thank the German Federal Institute for Risk Assessment, Hermann Broll, and the European Commission, JRC, Ursula Vincent, but closer to home at LGC, my team leader, Alison Wolford, for the molecular biology team, and other individuals, Gavin Nixon, Claire Bushell, Miguel Perdeguero, and Catherine Pay, the competent authorities in the UK, Food Standards Agency and DEFRA, ENGEL, clearly Guy van der Eede, but also the other chairs as well, Joachim, Hendrik, Ursula Vincent, and all other ENGL colleagues, and also my colleagues at SASA and FERA for very insightful and very useful conversations there. And thank you for everybody for your time today, and also the organisers for establishing this very useful conference. Thank you for your time.